codon could be A, G, C, or T. So 4 choices there and 4 more choices for the third. And mathematically, if you have 4 choices followed by 4 choices followed by 4 choices, you multiply the possibilities, and that would give you 64 possible codons. And here's a list of them all. And this is called the genetic code, and it's somewhere in your textbook. I'm not exactly sure what page it's on, but it lists all the possible codons, all 64 codons, and next to each one it tells you which amino acid. Leucine is an amino acid, isoleucine is an amino acid, methionine is an amino acid, valine is an amino acid, like that. Now, what's odd about what I'm showing you here as opposed to what I'm showing you there? There's a certain difference. You may see what it is. The what? The U's? Yeah, that this has U's instead of T's, and so these codons are for what molecule? RNA, because we have, remember, RNA has U's instead of T's, whereas DNA has T's instead of U's. And so it's really the same genetic code, but it's written in RNA language instead of DNA language. And the reason for that is, as I'll discuss shortly, when the cell actually gets to constructing the protein over at the ribosome, it doesn't actually use the piece of DNA itself. No piece of DNA actually exits the nucleus and goes to the ribosome. What happens is a temporary disposable copy of one of these strands gets made into an RNA molecule, and that RNA copy is what gets sent out of the nucleus to the ribosome. And so to acknowledge that fact, they usually write the genetic code in RNA codons. But the point here is that these same codons appear in the genes in the DNA, except that the U's are T, since we're talking about DNA molecules. Okay, so the first codon of this gene is ATG, and if you remember to change your T's into U's, here's the codon AUG, and so to the cell that stands for the amino acid methionine, and so that's how the cell knows that this particular protein is supposed to begin with the amino acid methionine, like that. The second codon of this gene is GAA, and to the cell, GAA stands for the amino acid glutamine, and glutamate, okay. And so that's what the second amino acid is going to be. The third codon is CCC, here, and to the cell, that codon in the DNA stands for the amino acid proline, so the cell knows that the third amino acid of this protein is the amino acid proline. The fourth codon is kind of special. In DNA language, it's TGA, which in RNA language is UGA, and that's what's called a stop codon. That just tells the cell, you've reached the end of the protein, no more amino acids. And of the 64 codons in the genetic code, there are 3 stop codons, UAA, UAG, and UGA. All the others stand for amino acids. Now, looking at the genetic code here, notice in many cases there are several codons that stand for one particular amino acid, like isoleucine amino acid can be UAA or AUU or AUC or AUA, and leucine amino acid can have several, UUA, UUG, CUU, CUC, C blah blah blah, CUG. And I guess that sort of makes sense because there are only 20 amino acids, but there are 64 possible codons, so we expect some redundancy, or I think the official term is degeneracy, but it just means that you'll have more than one codon for any particular amino acid. Anyway, so using this genetic code, the cell would understand, would know that this particular gene encoded a protein of methionine, followed by glutamate, followed by proline. Sound good so far? All right, now, oh yeah, the TGA is the stop codon. And so there are the 4 codons and the 3 amino acid protein that's encoded by this particular gene as written in its codons. Okay, so here is your chance to work with the genetic code. There's a gene there, and oh, I forget how I wrote this. I think this is the first codon right here. Let's go with that. So using that as the first codon, what's the first amino acid going to be? ACA is THR, which is threonine. What's the second amino acid going to be? AAA. And where is that on our chart? Here it is. Lysine is this particular amino acid. And then the third codon, TCG, which is going to be UCG here. Serine amino acid. And then, what's the next amino acid? Get ready for a trick question. Stop codon, yeah, so there is no 4th amino acid. TGA in RNA language is UGA. Here it is. And so that's a stop codon. So this should be a 3 amino acid protein, threonine, lysine, serine. I think that's what I said. And that's it. This is a 3 amino acid protein encoded by this particular gene. And here's another one, and let's not do the whole thing, but we'll just do the first amino acid. What amino acid is encoded by this codon? So it's going to be UUU, which is, there it is, phenylalanine would be the first amino acid of this protein. And there it is. If you do the translating, that's what you get. Yeah. The one, that was just my fault when I was making the diagram. I just, for some reason, didn't begin at the beginning of the diagram.
So you always start with the first one?
Yeah, so like whenever I show you, remember that these genes are segments of chromosomes. And so even though I'm not showing it, there's more nucleotides further on in this direction and more nucleotides further on in this direction. But I'll always point out to you where the gene's supposed to start along the chromosome. You won't have to guess which one's the first codon. I'll always tell you where that is. All right. Okay, now before we leave this genetic code, one of the remarkable things about it is it's universal among all life forms on Earth. It doesn't matter whether you're a human being or you're some tiny little microscopic bacteria. Every life form on Earth uses the exact genetic code to translate its genes into proteins. And that's kind of amazing because, you know, there's been life on Earth for around 3.5 billion years now. And we all use the same genetic code. And they think that means that probably life only originated one time on Earth. So we're all, in a sense, ancestors of the first life form. And this is the code that that first life form had. And we've all retained it. We've all inherited it. Now, one of the practical upshots of that is that we can now move DNA between different species. This didn't used to happen until biotechnology was invented, but they can take human genes and stick them in bacteria, and the bacteria will express those genes, which means to make their proteins just fine. They can take bacteria and put in, let's say, genes for the human protein insulin and manufacture insulin that they can give to diabetics, and that insulin is made by the bacteria. And I think I showed you this slide before. They took some genes for some human proteins and put them inside the DNA of this goat. Well, the goat uses the same genetic code, so it makes the correct human proteins, and they milk the goat and then purify the human proteins away from the milk, and you get, again, pharmaceutical drugs. And this is one of my favorites. Remember, this one is where they took the gene from a firefly that allows it to glow in the dark, and they stuck it inside a plant, and plant uses the same genetic code as the firefly, so you get a glow-in-the-dark plant. Just mind-boggling. Okay, so the genes encode the amino acid sequence of each protein by these codons, these 3-letter series of nucleotides. And you have around 25,000 genes, which indicates that you have around 25,000, as a human being, you're capable of generating around 25,000 different proteins. Now, later on in the semester, I'm going to change what I just said slightly, because it turns out there are certain ways you can get more than one protein out of any particular gene. But for right now, we'll say that human beings can make at least 25,000 proteins because they have 25,000 genes. Okay, well, getting back to this right here. So the gene there in the nucleus holds the recipe for the protein as written in its codons, and the ribosome is floating around outside the nucleus in the cytoplasm. And so the ribosome needs that genetic information. It needs those codons to know which particular amino acids it's supposed to link together to make the protein. And there's going to be 2 steps in going from the gene to actually making the protein, and they're called transcription and translation. And here they are in a nutshell. In transcription, a temporary disposable copy of the gene is made that's called a messenger RNA, or mRNA for short. And that RNA copy of the DNA gene is sent out of the nucleus. Notice the nucleus has little holes in its membrane, and goes to the ribosome out in the cytoplasm. And then using the codons in that mRNA as a guide, the ribosome will then link together the correct amino acids to make the protein. Go back. And that process of the ribosome actually reading the codons on the mRNA to link together the amino acids is called the translation step. So transcription, copying the gene into RNA, translation using the RNA to actually make the protein. Let's go back to that gene inside the nucleus and see the transcription in a little more detail. All right, so there's our gene. 2 complementary pieces of DNA, it's part of a chromosome. For transcription,